Well, yesterday I entered La Rioja on the N111, and so I saw what a special place I was coming to. It was just spectacular. I'm really very pleased to be here, and thanks very much for the invitation to speak today. And it's good to be here with Raquel again, because Raquel was in my lab for close to 4 years at least, I think. So, does anybody know what the artwork is here? Anybody know what all of that comes from? Those are the labels that they used to put on packing boxes for citrus, okay? So last week I was actually harvesting Valencia oranges in Florida. So I've worked with citrus now for over 30 years, and it's about all I know anymore. Today, though, what I'd like to talk about is the idea of conservation biological control. I'm a nematologist, and so, you know, all of my work takes place below ground, essentially. And what I wanted to talk about is the possibility of doing conservation biological control that's subterranean rather than what we all know about, what a lot of you are studying here, as a matter of fact, conservation of natural enemies above ground. It's one of the most important aspects of pest control for many, many years, but subterranean pest control, that's a little bit iffy because it's difficult to see those things. And until very recently, we just didn't have the tools we needed to have a really integrated look. So that's what I want to focus on today. And essentially what we've been asking for a number of years now, for almost 20 years, is what natural enemies occur in the soil for the system that I'm working on. Where do we find those natural enemies, and why? Why are they in some places and not in others? And then how effective really are they? Are they really worth paying attention to, and are they worth considering conservation biological control? So this is the system I work on. This is a weevil that came into Florida about 40 years ago. It is a horrible pest. It causes this kind of damage to the root system of the tree, so you can see that it can kill trees very easily. It interacts with Phytophthora, so it's a pest disease complex. The name of this weevil is Diaprepes abbreviatus. We actually have a complex of about 4 weevils, but this is really the bad one. Diaprepes adults live in the canopy of the tree where they lay their eggs, and when those eggs hatch, they fall to the soil. And the larvae spend anywhere from 3 to 6 months in the soil, growing larger and larger, from almost microscopic size to a very large weevil larvae here. And as they get larger and larger, they cause these channels in the cortex of the roots until when they get up in the crown area, they can kill the tree. So it's a very serious problem. The one thing that growers, the ways that growers have tried to manage this relatively unsuccessfully all these years, I mean, it's still a really big problem, is to use pesticides above ground to try and control the adults and to try and control the eggs from hatching and things like that. But in Florida, our soils are so sandy that you really can't put pesticides on the soil. They'll just end up in the groundwater. So for over 20 years now, they've used entomopathogenic nematodes, insect-killing nematodes, that I'm going to just call EPNs from now on, to try and help manage, you know, in an IPM program, to manage these grubs in the soil. And the nematodes really are quite effective. So just real quickly, here's what these nematodes do. They start off in a product that can be made commercially in bioreactors in very large quantities. Doesn't cost very much money, maybe $30 an acre. I didn't translate it into euros, I'm sorry. To treat with these each time they do a treatment. When they come out of this bucket, they're 3rd stage juveniles. That's the survival stage. That's the stage that moves through the soil. It encounters an insect and it infects the insect. And these nematodes live in symbiosis with bacteria, with entomopathogenic bacteria. All right? So once they're inside of the insect, they release this very specific associated bacterium that they carry. It's either a Xenorhabdus or a Photorhabdus species, depending on the species of nematode here. The bacterium kills the insect, and then the nematodes and the bacteria reproduce for a couple of generations, nematode feeding on the bacteria and on the tissues that are being degraded in the insect. And then eventually, as many as 100,000 new nematodes might emerge from that cadaver. And off into the soil they go to serve as food mainly for other organisms, but also to try and infect other insects. Now, when I first started working with this, we wanted to know how effective are these entomopathogenic nematodes, these commercial nematodes. So the way that we looked for that is to put a larva inside of this little cylinder here that's got a mesh screen on it. Ants can't get in, but nematodes can. And so we fill this with moist sand, we put an insect in, we put a snap cap on both ends, and then we bury it in the soil. Here you can see these guys burying it in the soil. And we leave it there for, we do our treatment after that. We spray our nematodes on the soil surface. A week later, we pull the cages up. We see whether the insect is dead. And if it's dead, we put it on what's called a white trap. And that's just simply a method of incubation to wait and see if nematodes come out. Was it nematodes that killed them? And if it was, what nematodes killed them? And that's where the real surprise and the real adventure began for us, because we were spraying these things on and we were seeing mortality of up to 90% of the larvae within a week. But the nematodes that were coming back out weren't the nematodes that we were necessarily putting into the soil. We were finding lots of different undescribed species of entomopathogenic nematodes, as well as a couple of described species. And so that's when we began to look at what's happening to the communities in the soil and looking at the food web. Now, the way citrus is grown in Florida is it's grown in two what we call ecoregions. There's a central ridge of deep, deep, deep sand that runs down the middle of the state. And the rest of the citrus is grown in an area that's very shallow soils with the groundwater very near to the surface. So this is a relatively dry region, not so much unlike your Mediterranean situation here. And this is a very wet situation. And citrus is grown in both of those areas. We call this the central ridge, and we call this the flatwoods. Okay, and what we knew is that Diaprepes, the weevil, is a big problem in the flatwoods, and not so much of a problem on the central ridge. And when we started to do our work on EPNs, we found that the naturally occurring EPNs were very, very effective on the central ridge, but not in the flatwoods. Okay, so the obvious question was whether or not the EPNs were regulating the weevil. And if they were regulating the weevils, then we wanted to know why were they so effective here and not effective there. And that's more or less the rest of this story. So it's a very simple model. This is a trophic cascade where the endemic nematodes, the native nematodes, are controlling a pest, and they're having a big effect on the citrus tree, we hypothesized. And if this is true, then what is causing them to be very effective in one region and not in the other? Is it mainly the physical properties of the soil? Or is it natural enemies that might be affected by the physical properties of the soil and be more in one area than another? That's essentially what this talk is about. So 10 years ago, 20 years ago, really, when we started doing this work, the tools that we had were, were a little bit less than what we really needed. We would extract nematodes by just centrifuging them from the soil in a sugar solution. And if we wanted to look at the fungi that are in the soil that might be affecting this system, we had to do dilution plating, all right? And if we wanted to look at mites in the soil that might be eating the nematodes, things like that, we would have to use Berlese funnels. And, quantitation was very inexact. We could not discriminate with these these nematophagous fungi. Here's a fungus trapping a nematode. Here's a fungus that has infected a nematode with zoospores. We couldn't discriminate whether or not these were feeding on organic matter in the soil, because they do that. They're saprophytes, but they can also kill nematodes. So we really didn't know, are they killing nematodes? Are they feeding on other things? And also, if we culture them, you're on a wet petri dish, it's going to favor an organism like this over an organism like this. It's going to bias what we're seeing anyway. Today, obviously, things have changed. We're primarily using quantitative PCR with this sort of a method. So now what we've learned is that all we've got to do is sucrocentrifuge the nematodes from the soil. That's all we need to do. If we pull the nematodes out the way we typically do, we're also going to get mites, we've learned. We're going to get more mites than you get on a Berlese. And we're only going to be getting fungi that are associated intimately with the nematodes. So it's a little bit better ball game from that standpoint. Much lower detection limits for all of these organisms. As I mentioned, we're probably detecting nematophagy better than just saprophagy with this method. The only drawback that I really see is with qPCR, you only see what you're looking for. You have to have the primers and probes, and you're only going to find what you probe for. So that is a major drawback. And plus, you're not looking in the microscope so much anymore. You don't see some stuff that I'll show you in a little while. So what I'm going to talk about primarily is a few surveys that I did with Raquel. Raquel came and provided all of the tools we needed to do all of this stuff. We did geospatial surveys, both in citrus orchards initially and then in natural areas. This is the number of sites that we sampled in. And we did 4 temporal surveys over a couple of year period that I'll show you some data as well. And what we measured then in these surveys were 13 species of entomopathogenic nematodes and some natural enemies of the nematodes that we had found were important in our earlier work. Free-living nematodes that compete very well for that cadaver. Ectoparasitic bacteria that you can see here that I'll talk in a little bit about. And nematophagous fungi, 7 species of those, as well as just about everything that you can think of that you could measure in the soil that might be affecting these organisms. And we took a couple of samples from each site, extracted the nematodes, got the DNA, and that was it. And after all of that work and all of that time, we didn't see a lot of relationships with the natural enemies except positive associations. The kind of associations you might expect to see if you had a lot of EPNs, that you're going to see a lot of their natural enemies. But what we did find is very strong relationships with things that primarily influence the soil moisture. And nematodes are aquatic organisms, so it made a lot of sense. But the depth of the groundwater was very important, the water holding capacity, and the organic and clay content of the soils. All of those things help to describe the properties that we saw on a geospatial basis in this survey. So essentially, what we learned from all of that is that these physical factors seem to be the most important determinants of where we're finding the different nematodes. Let me show you the nematodes we found, though. It's interesting. Essentially, on the ridge, this is just some typical data. On the ridge, we generally found 2 or 3 species co-inhabiting, and we found them in 100% of the samples. All right? So here we have a species called Heterorhabditis zealandica. Here's S. diaprepisi, H. indica. This is a Steiner nematode. There's only 2 genera of these nematodes, Heterorhabditis and Steiner nema. So you're going to see that a lot. So we would see maybe 3 species, and this is just at the same time, they're just sort of different parts of the same field. And then in the flatwoods, here's a couple of flatwoods orchards, we would either see a lot of H. indica all over the field, or occasionally just a clump of H. indica, just very, very few nematodes in a 20-hectare area, for example, in this particular case. There were other nematodes in the flatwoods, but you would see them very rarely. Actually, I show you here. On the central ridge, we always found this nematode. This Steiner nema diaprepisi, SD, is what I think is the most important nematode to have on the central ridge. We found HI, Heterorhabditis indica, on the ridge or the flatwoods, and occasionally we would find this Heterorhabditis zealandica. In the flatwoods, it was a very different community. As I said, H. indica everywhere, 100% of the time. SX, we now call it Steiner nema kungai. It's got a name finally. I still prefer SX, to be honest with you. SX, occasionally, and I consider this a very important nematode in the flatwoods. Unfortunately, you don't find it everywhere. And occasionally SD, but very rarely. Okay, so SX in the wet areas, SD in the dry areas. And when we came back in to look to see whether or not those, the relationships we were seeing, the spatial relationships made sense, we looked specifically at the SD and the SX, the 2 nematodes that inhabited very different ecoregions. And we found indeed that if we had a column with moist soil at one end and dry at the other, the SD would migrate preferentially to the dry end and the SX to the wet end. And if we put them in glycerin, here we put them in a very high rate of glycerin that's going to dehydrate these nematodes, The SD survived that over a period of time nicely. There's a couple of populations here and a couple of populations of SX that just simply don't survive desiccation the way that the SD does. And it's the same thing for survival in sand over time. A couple of populations of SD are surviving nicely in the 6% soil moisture, not in the 18%, whereas the SX surviving in the 18%. So we've got these nematodes that physiologically are very different. And when we looked, I won't belabor these things, just to point out that some of these nematodes were better than others at protecting the plant. And it was always the Steiner nematodes. Okay, so these two, then they're large nematodes, these two big nematodes, SD and SX, in greenhouse experiments, if you're looking at root weights, for example, you can see how much root weight we had on a Heterorhabditis indica, which is everywhere, but it wasn't protecting to the same degree that this SD and SX were. Forget about SR, that's an introduced nematode. That's a commercial nematode. It's not a naturally occurring one. And if you look at in those experiments where we had put diaprepes in the planting, in the container, and then treated with the various nematodes to see what would happen, we found that the diaprepes adults that finally emerged from here, that there were far fewer for any of them that had the SD or the SX than these other treatments, either the untreated or the Heterorhabditis. So over time, we've become very convinced that you want to have certain nematodes in your community to be effective in the field. And so these are just some of the things that we recommend to growers that we consider important in terms of conservation of what's happening in the soil. And I'm only going to talk about a couple today because I was told I only give 40 minutes. I'm joking. So anyway, let me just go on to the first one. It was real obvious to us, okay, they're working well on the central ridge. They're not working so well in the flatwoods. So before we knew very much about these nematodes at all, we said, well, let's go to the flatwoods. Let's plant some trees, but let's bring sand from the central ridge and put it in the planting hole and compare that to where we don't do that. And so that's what you see in this experiment. In this experiment, this went on for about 6 years at least. We would trap the diaprepes adults emerging from the soil. We would bury these larvae from time to time to see what's happening and use the qPCR primers and probes as well to see how many are there. We looked at these things for quite a while, and essentially what we found, and this is the kind of damage that you can see on the leaves of the tree from the diaprepes adults, by the way. And what you can see here is where we have sand in the planting hole. This isn't, I didn't just select these two trees. This is pretty typical. Here's sand in the planting hole versus no sand in the planting hole. A great deal of mortality here. Whoops. A great deal of mortality here, almost no mortality of the trees here. And essentially over time we found that there were far fewer weevils emerging from the sand. We had much more active EPNs when we would bury our larvae or we would check them with qPCR. Tree survival was better, as I mentioned. And I didn't mention that we inoculated all of these plots with the various species of the EPNs as well. And so because this grove actually only had HI in it, the indica, and not very much indica to begin with. So we inoculated all of these plots initially in the experiment with the nematodes. And at the end of the experiment, you see what happened in the sand and the native soil here. At the end of the experiment, most of the nematodes that we found were Heterorhabditis indica or Steinernema diaprepesi, a lot of Steinernema rio brave as well, the introduced species into Florida, excuse me. Very little SX. And this is a flatwoods grove. This is a grove that was really a wet grove. That's why we chose it. But very little SX at that time. And in the native soil, no SX and pretty much the same thing, SD and HI. But a couple of years later, a student came down and wanted to learn how to do the qPCR. So we said, well, let's go back out there and see what it looks like. And when we did it, we found that indeed, 2 years later, the dominant nematodes in both the native soil and in the sandy soil were SX and HI, which you would have predicted based on their habits, where we find them naturally. So that's why we recommend that you introduce SX into these plots where you don't find it, or into these fields where you don't find it. as well as a lot of growers do put sand in their planting holes. Not necessarily for this, but for a lot of different reasons now to help with drainage and that sort of thing. Another thing that we recommend is that they reduce the soil pH. And this is fortunate for us because for other reasons we have citrus greening, a very serious disease in Florida, and we've learned that citrus trees don't tolerate high pH when they have that kind of stress. So they're reducing their pH as well, but they're getting some additional benefits that they may not even realize. I told you that there was a bacterium, an ectoparasitic bacterium that we discovered a long time ago on the surfaces of Steinernema diaprepisi, all right? And this just shows you the endospores here on the cuticle. And essentially what this bacterium does is it just adheres to the cuticle, gets inside of the insect with the other bacterium and the nematode, and it reproduces along with the other two organisms. And it just rides on to the next insect. So it's just a phoretic, phoretic exploitation that's going on. Here's once the nematode gets inside of the insect, it casts off its third stage cuticle, and this is just the empty cuticle, and this is one that didn't have any spores on it, and here's one that did have spores on it. And you can see that very quickly those spores germinate, and you end up with these rod-shaped, it turns out they're pennibacillus that are forming in there. And the pennibacillus, we've determined were not entomopathogenic at all, but they are very closely related to some of these pennibacillus species that are entomopathogens, all right? So they probably were entomopathogens that eventually learned they didn't have to do that. They could just hitch a ride, and that's more or less how they evolved, I imagine. But they're not entomopathogenic. They are specific to S. diaprepisi for the most part. They're primarily species specific. They did not inhibit the EPN reproduction whatsoever. All they did was impair their ability to move through the soil. They moved slower, and so they didn't find hosts as well. And so we always wondered, well, do they have any role in regulating these nematodes in nature? We just had a hard time figuring that one out. We got some clues, though, when we worked in a system that was being developed to make the trees grow faster. It's called the Advanced Citriculture system. And essentially, all that was happening here is that these trees were being fertigated every day. So they were being given nutrients and water every single day, as opposed to the conventional citriculture where you put a little dry fertilizer out 3 or 4 times a year and irrigate with micro sprinklers as needed. Here they were getting drip system, so it's a concentrated wet area daily. So the conditions in the soil were very different, and so that seemed like an excellent opportunity to go and see what's happening to your nematodes. And when we did that, we found that indeed, in the advanced citriculture system that had wetter soils, we found fewer S. diaprepisi, and those S. diaprepisi were infested with more pennibacillus spores than in the conventional citriculture. And among many, many things that were different in those systems, you know, chemically speaking as well as physically speaking. pH was very much higher in this where we had the problem with the S. diaprepisi, and so we looked at that first. And we were just lucky because it turns out that if you put spore-encumbered nematodes in a solution at different pHs, that at a high pH, they retain those spores, but at a lower pH, the spores fall off. They won't adhere to the cuticle. And so, when we found that that was the case and that we had differences in the populations of the nematodes, we continued to study that to see, well, is that actually having any effect on pest management? And in fact, we saw that the Diaprepes abbreviatus that happened to be in that grove were much higher where they had the advanced citriculture system and the fewer SD than in the conventional citriculture. This is another weevil. I told you it was a complex in Florida. Also, there were more of those weevils where we had the advanced system. And indeed, we even had more Phytophthora. If we control the Diaprepes, we actually reduce the Phytophthora as well. So we had pretty good evidence that there may be this association that we're seeing with the bacterium may actually be having some effect there. But what I'm going to show you now is we actually were able to see that a little more clearly, I think. So the one thing that we were really hoping to see is sort of like the tsetse fly, why you don't have horses in East Africa, you know, because you've got this pest there that would kill the horses. We were always looking for natural enemies that were in the flatwoods, maybe, that were describing why we didn't have so many species of entomopathogenic nematodes in the flatwoods. And you know, we weren't seeing a whole lot in these geospatial surveys and the spatial patterns that we were looking at. But on the other hand, if we're going to look at something, we want to look at flux. If we want to see predator-prey relationships, then we want to look at a temporal relationship. So we had those other studies that we had done. Essentially, in those studies, we had four different groves where we looked, or I should say sites, because two of the sites were in the same grove, at just the same 10 trees for 2 years. All right, we would take 3 cores of soil from each tree, and we'd composite these things, and walk out with essentially 3 samples from each one of these groves each month. And we divided those samples between as far as being deeper or shallower as well. And we did the same thing we did with the others, except of course we didn't measure so many soil properties this time, because those things aren't going to change over time, except things like pH and cation exchange. And essentially what we saw in those 4 sites, and here I'm showing what happened top and bottom in the deeper sample and in the shallower sample. What we saw was what we had seen in the previous experiment that I just showed you. There was a difference in the pH across those sites, and where we had higher pH, we had fewer Steinernema prepuzii. And those Steinernema prepuzii were infested more heavily with the Penibacillus bacterium. And what I thought was really interesting is when we finally analyzed the entire data set, one of the things we were able to see is this sort of Lotka-Volterra relationship that probably a lot of you remember from your beginning ecology classes, where if you're looking at the relationship of the predator population compared to the prey population, you know, as the prey population is increasing, then the predator is increasing up to a point. When the predator gets to be too numerous, then the prey population starts to decline. And so you've got this counterclockwise temporal cycle going on with your oscillating curves, you know, first your prey and then followed by your predator going through the system. And we saw that in all, the Steinernema prepuzii, which can have the Penibacillus, was in 3 of the 4 sites. And in all 3 sites, whenever there were major changes in the population density of the nematodes, you get this counterclockwise movement. This is really clear in here, where you just go like this all the way around, month after month. Anytime we had these kinds of things, and they were very, very significant, even over the whole time course of the experiments, that we're getting a counterclockwise movement in the relationship between Penibacillus and the prey population. So we think we've got pretty good evidence, really, that Penibacillus does regulate Steinernema prepuzii, and that we can affect that by affecting the pH of the soil. And this is just, I'm not going to belabor this, but see, these are some of the other natural enemies, and we were finally starting to see some nice relationships that you would expect to see if they are actually regulating the nematode populations. We've got some inverse relationships of a free-living nematode that we know competes with. These are the 3 Steinernema sites, and we've got really consistent relationships here. This is a very, very this is a fungus, this is Arthrobotrys dactyloides. It's a very nematophagous fungus. It relies on nematophagy more than these other guys. And in fact, we're seeing some really nice relationships with the 2 heterorhabditis sites as well. So we're seeing some nice information there as well. I'm going to finish up today by just showing you some things that I think are really cool. And these are all projects that were done by graduate students at CREC over the last 6-7 years. So essentially what all of this involves is chemical ecology. Now, Lucas Stalinsky came to CREC, was really interested in looking at chemical ecology. not just on the basis of insects, but also nematodes. And he had a student, I'll show you his work in just a minute. But essentially, we're learning more and more that EPNs really communicate effectively with lots of organisms, including plants, in the soil, and that we will, to me, this is really where we want to be looking if we want to think about techniques, tactics for conservation biocontrol. So we're looking at exploiting anything we can learn about this right now. And the example that I think of is like the push-pull system, where entomologists try and interplant crops that might push pests out of an area and maybe pull them with other plants into an area on the edges of fields, attract natural enemies as well. These are the sorts of things that we'd like to be able to do in the rhizosphere as well. So these are some olfactometers that are kind of the basis with nematodes of how we're going to know whether something's attractive or repellent or whatever. And so we can, for example, here, this is the first example I'm going to show you. Lucas's student, Jared Ali, let Diaprepes weevils feed on some of these plants and not on others. And then he would put the nematodes in the middle of the pot here, and 24 hours later he'd see which ones of these ends he would find them in, all right? And these basically do the same thing, just a lot cheaper equipment. It's very expensive. This is what I prefer. All right, so what Jared found is that when the weevils are feeding on the roots of citrus, the citrus is producing a compound called pregagerine. And pregagerine is fairly rare in most plants, but citrus give off this, at least some citrus give off this, when they're fed upon by the Diaprepes, and pregagerine attracts entomopathogenic nematodes. And so even when he went out into the field with larvae here that you can see in buried cages, maybe put a little pregerine in there or didn't put it on them, he found that in fact the larval mortality was much higher when he put that volatile along with the larvae in the soil. And this is just showing you, I believe, let's see, that's the larval mortality, and this is the numbers of EPNs that he found either in the cage or on soil that had adhered to the cage. So one of the things that we recommend to growers, because he also went to screen the germplasm to see what most of the rootstocks do, and for example, one of the widely planted rootstocks worldwide is sour orange. It produces pregerine constitutively. It just always emanates from the roots, so we don't really see how that's going to be of any use. However, trifoliate varieties like Swingle citrumalo, one we use a lot in Florida, they only produce it when they're being challenged by these or fed upon by these insects. And so there we've got something that presumably is going to be beneficial from the standpoint of attracting entomopathogenic nematodes. This is an interesting one that we're working on right now. I had a student, I guess 4 years ago now, just showing him how to do these surveys. He went out and he buried 200 actually, insects all over a grove, and he found half of them when we plated them out here looking at the cadavers to look to see what nematodes were coming out. Instead, fungi were growing on them, on fully half of them from all over the field. And so the fact that we know that a lot of free-living nematodes compete with EPNs, EPNs will kill the insect, but then the free-living nematode gets in and co-opts the situation, and it reproduces instead. The same sort of thing might be happening with this fungus, we thought. So we studied that, and it turned out it was Fusarium solani. And in his controlled experiments, Cheng-Yen Wu showed that if we had Steinernema diaprepisi by itself or combined with Fusarium solani in the system he was working with, he killed more insects with the Fusarium solani present, and obviously produced more nematodes with the Fusarium present. And he used these kinds of assays, so he would have the fungus and the insect over here versus no fungus and the insect over here, see which way they were going. He also showed, what was I looking at? I guess it's the same thing. He also showed that the Fusarium population increases more in response to that situation as well. So for Fusarium, here's an insect that's being killed by this nematode, and I'm either going to compete with the nematode and get the whole cadaver, or at least there'll be something left over for me. But in that situation where both were present and the larva died, here's where the larva stayed alive. Here's Fusarium aloum, Fusarium with the S. diaprepisi. These are on live larvae, versus where it died, the Fusarium population was much higher. So it's a mutualistic system, but the question is, how did it evolve? And again, he did the same thing that Jared Ali did. He looked to see what volatiles were being produced in this system. And here's one that was being produced by Fusarium solani that attracts the EPNs. It wasn't really produced by the Fusarium oxysporum, which does not attract the EPNs. And this happened to be 1-octatriol. And 1-octatriol is replete, and the literature is replete with articles showing that a lot of insects are attracted to that alcohol. It's produced by mushrooms, so things like fungus gnats and that sort of thing are attracted to it. And so that could be the situation whereby you've got an insect being attracted to the Fusarium solani. It's beneficial for the insect in a sense. The Fusarium solani is also attracting EPNs. And so you've got a system here, and this is just showing you what it looks like in another kind of an assay. After 19 hours, this is where you had the 1-octatriol, and this is Steinernema diaprepisi versus the control. And so it's very attractive to S. diaprepisi. It was totally repellent to Steinernema diaprepisi. But Steinernema diaprepisi doesn't feed on fungi, and probably has a good reason not to want to go toward 1,3-octanol because of this sort of a situation. So you could be having evolution occurring in both situations, but what we're really interested in right now is screening for insects that are and there are a lot of volatiles that were coming off that were attractive to EPNs, not just this 1-octatriol. So what insects are attracted to those things at the same time that EPNs are? And now we're really thinking of a system where we're talking push-pull, where we could maybe have pellets of this compound that is attracting an insect and an entomopathogenic nematode, keeping it away from the roots, leading the insect to its doom, benefiting the EPNs. It could be a really nice system to continue to look at. But we're just starting on this one. And these are just two more. One of them that I think is really neat, just to show you some of the things that the students have been doing. You spray the top of the citrus seedling with salicylic acid, and the root system is attractive suddenly to entomopathogenic nematodes. And the compound that's coming off that's probably responsible for that is D-limonene that was coming off in response to the salicylic acid that was being sprayed on the leaves. And this is the last that I'll show you. And this is the neatest of all. This is essentially showing that these little guys learn really well. Some of the terpenes that we expose these nematodes to are repellent. Some of them are attractive. This pinene compound was repellent. Anything that's below 50% is a repellent. Anything above is attractive. Was repellent to both Steinernema diaprepisi and H. indica. But if you incubate them in a dilute concentration of that for 24 hours and then expose them in a t-test, t-tube test, they both become attractive. All right? They're both attracted to it. And the neat thing is that this is the control again, just like this. If you expose just the S. diaprepisi for 24 hours, still both of these species are attracted when they're exposed 24 hours later. They're communicating with each other, and I just think that's so neat. And what's really neat about this too is, think about how simple the nematode physiology is. It's got 302 total neurons in it compared to all the billions of neurons that we have when we try and learn something. So I think, if I'm not mistaken, yeah. So the only thing I want to conclude with is that I've shown you that I think entomopathogenic nematodes are important in this diaprepi situation in Florida, but I don't really know how common that is worldwide, because one of the things that we learned over this period of time is that what we report is rather unusual. When we sample citrus groves, every time, 100% of the time, we find entomopathogenic nematodes. Other studies reports for many, many years is closer to 10 or 15%, and so the question is it Florida or is it the methods? because we were pretty much the first people that were looking at it in the sense of using qPCR. So Raquel came to Portugal sometime after having done this work in Florida, and she saw somewhat similar results looking at a lot of different habitats in Portugal that she saw in here. She did see a lower detection frequency, but not that much lower with her methods. The neat thing is that the similar sorts of properties in the soil of all the things that we measured were responsible for the patterns, were mostly responsible for the variation of the patterns that she saw. So she was seeing very similar results to what she saw in Florida. This is just typical. I could show you probably 100 papers that show you the frequency of detection of EPNs. This is typical. This is occasional, as high as this. This is what Raquel saw in her natural areas. Here's what we saw in Florida in the natural areas. Here's in the citrus groves. But these are the same samples from Portugal that Raquel sent over to a guy now in Florida who's using next generation sequencing to look for these organisms. And this is what he found. All right? So it's something very different. And qPCR was wonderful, but like I said, you only see what you're looking for, and with NGS, you see whatever you want to see. You see what's there. So that is really exciting. And so, we're at a point right now, I think, where we can see worldwide what's there, where are we finding these different species, why are we finding them there, what are they really related to, what's causing the patterns that we see, and then finally, how effective are they? And so, of course, you recognize Raquel. These are the other people. Fahim El-Borai, Ectopatak, did the work with the nematophagous fungi that I showed you, most of that work. Robin Stewart is a great colleague and did a lot of this work as well. This is Josh Flutie, who works in the laboratory. Currently, Shengyan Wu is doing the work with the Fusarium solani, and Alexandros Dritsulis is working with the NGS system right now that we'll be looking at. So, thank you very much, and if you have any questions, I'd love to try and answer them. Yes.
You mentioned that the detection method is really important. Did you try in your lab, do you know droplet digital PCR? If you try this kind of PCR to the detection of nematodes and microbes?
No, just tell me a little bit about why.
It's another step forward.
Mm-hmm.
After qPCR, it's also detect,
Yep.
quantify,
Yeah.
but they don't use standard Qs.
And
also suppose the inhibitors are less harmful.
Yeah.
For the method. We are trying to compare to PCR and
Yeah.
droplet digital PCR to see if it's more efficient or not.
I guess we did discuss the possibility of doing that. That shows you what my memory is like these days. But yeah, I think that's great. We were really excited about that. But now all I can think about is next generation sequencing. Yeah. Oh, absolutely. Oh, you can. Yeah, that's one thing that we didn't succeed at all with that. Yeah, that would be terrific. Absolutely. Yeah. Yeah. Yep. But I think though that very possibly, if you're talking about controlled conditions, controlled experiments, that system might be the one you'd want to go with for sure. Where you, yeah, as long as you know specifically what you want to be measuring, I think that'd be excellent. Yeah. Yeah.
A little bit expensive, but now, maybe later. So you don't have money or...
I never succeeded in getting Raquel to stop telling people that it was faster and cheaper than the methods we used to use. It was pretty expensive at the time. It's getting a lot better though. Yeah. Any other questions? All right. Oh, yeah.
Well, I have a question regarding seems to be quite effective the use of EPNs in the type of crop. However, probably has problems in the way it changes the ecological systems of the soil. You require using it constantly. What is your impression?
Yeah, well, actually, I would say yes and no to the question of whether it's highly effective. When we have taken had the opportunity to treat over a number of years and capture the number of adults coming out of the soil, essentially what we find is that when we put these nematodes on the soil, they kill up to 90% of the larvae in 1 week. And remember, these larvae are in the soil for months, all right? So they'll kill them really quickly. But the growers can only afford to do this maybe once or twice a year. And it's just like using a non-persistent pesticide. And these adults in the canopy, they're laying eggs that are dropping into the soil all year long. So what we're killing are being replaced very quickly. And the nematodes that come out of those cadavers are being very quickly picked off by mites. I mean, you can just see what's happening. They work for about 2 or 3 weeks very well, and then it's over. So 50% is about what we manage to reduce the population of adults by if we're just using EPNs over a period of time, twice a year. That's really not enough. We're working on systems where maybe we could just focus on the crown area of the tree, so we use fewer nematodes and we can just keep putting them all the time. I can't remember if I've meandered a little bit away from your question, though, the point of your question. That's essentially how effective I find them right now. If we could use them frequently, I think they might be the entire solution. We might not even need adulticides, but it's the cost.
Maybe I need the explanation, but I was surprised by one of the records you mentioned before, or in the beginning, about recovering different nematodes and from the larvae from those you use for inoculation.
Okay.
What was the explanation for that?
Okay. So actually that's what started all of this work. I would have been done once I saw how good the product worked and reported that. I would be going back to citrus nematode and burrowing nematode and other kinds of plant parasitic nematodes. But what we found when we bring the nematode back up, you saw in the dishes we had some of those cadavers there that had the fungus growing. Well, a lot of times just nematodes come out. And so what we were doing was we were saying, okay, we've got this much mortality in 1 week. Now let's make sure that the mortality was caused by the nematode. So we incubate them. Here come the nematodes. And we would look at them microscopically. And we found very quickly that, the nematodes that are coming out of here are, number one, they're free-living nematodes. They're not entomopathogenic. Or they're entomopathogenic, but they're not the species that we put on the soil. And so that's when we started to study the entire system to see, well, what are all of these different species of nematodes? And what about these free-living nematodes? What's that all about? Because if we just bury something and we don't spray any nematodes, we don't get any dead insects with free-living nematodes. It's only when we spray EPNs that we find these free-living nematodes. So clearly, the EPNs are killing the insect, but there are some free-living nematodes that are very well adapted to coming in quickly and competing with them, and sometimes out-competing them. Yeah. Well, thank you very much.